Hello everybody, welcome to another small lecture that's being used for the flipped classroom sessions in the biology program here at HEC. In this case, we're going to talk about an absolutely amazing group of organisms known as the Protists. This is a bit of a difficult group to talk about, if for any other reason, because the taxonomy with this group is quite messy, unfortunately. This is the group of organisms that biologists kind of had difficulty classifying. So what ended up happening was they kind of take all those organisms they didn't know what to do with and kind of stuck them in here. It's a bit like having, say, a junk drawer in a kitchen. I've actually got a couple of them, but it's that drawer, you know, that you take stuff you don't know really what to put and just say, oh, let's chuck it in there and hopefully that'll suffice. That's kind of what happened here. And because of that, we have all these organisms that are kind of related to not only maybe themselves, but to groups outside of this kingdom, and it becomes unwieldy fast. So we've had a lot of discussions in biology exactly who should be in the Protists. Should we remove them, put them into another group? Like for example, if it's alga, should the alga actually be here, or are they really plant-like, or even plants for that matter? If they're plants for that matter, maybe they should be in the kingdom Plantae. It's just a mess, and that's just with something like the alga. So unfortunately, this has become a group of organisms that are not monophyletic, and that's really where the problem lies with it. So as I mentioned to you before, as I go through this, I'll try my best to give some of the more current taxonomy that's here. But because the taxonomy is difficult, it's really one of those, wow, we kind of have to look to see today what's happening with the taxonomy. Has anything been removed? Has anything been added into these groups? And so the taxonomy can change really quite rapidly, and it's difficult to keep up with it then. With that, it should be noted that because this group is so unwieldy, there is sort of like a movement to disband the kingdom, actually get rid of it. It's kind of like the old Monarans, where you had the prokaryote, and even to some extent the viruses in it. And the viruses, of course, got booted out into their little world, and the prokaryotes pretty much got broken up into two main domains, of course, the archaea and the eubacteria. So this is kind of running down that track, where since it doesn't really work out to have a kingdom for the Protists, this group is being disbanded out and sort of broken up into all these different areas. So I'll give you a quick view of what the current taxonomy is, but like I've said before, don't ever really get married to anything in biology. It's really one of those, you have to be willing to change, and it's going to happen a lot in this field. So with that in mind, let's give this a roll. Because there's so many organisms here and so much to talk about with this group, it's really one of those chapters where I've kind of like got to say, well, here's the main idea, here's the main organism, here's kind of what it looks like, here's maybe some tidbits on a few organisms here and there, and kind of move on. If not, I could easily be here for 3-4 hours talking about this amazing group of organisms, and that's just a little bit outside of what we're trying to do. So it's a bit of a snapshot. As always, then I do recommend that you read the full notes that I have posted for you, as well as your textbook. Make sure you read, again, through the currently released edition that we're using to get, again, more of the taxonomy and more of the general idea that's current. And then, of course, ask any questions you might have. So with that in mind, let's go ahead and take a look at this. Now with that, it shouldn't be surprising to you then that if there's a lot of different kind of organisms that are in this group, that these guys are pretty much everywhere. There's some that are terrestrial, there's some are freshwater, some are marine. I mean, there's amazing. Some are solitary, some are colonial, some are pretty tiny, so we're talking like cell-like. Others are massive, massive structures. It could be like 100 feet long. It's really just quite amazing what's here. With that, though, obviously it's a really big protistan. You don't need a microscope to see that. So one of the more enjoyable things, and a little bit frightening to some degree, that I've done. I've been scuba diving since I was a child. I started when I was actually 9 or so. So I've been doing it both for fun, yes, but also for work for as long as I can remember. And one of the more interesting things I did, which again was a little bit spooky, was diving in California. Now the area I was in Southern California, so it's kind of chilly even down there, driving off places like the Costa Mesa areas and say Huntington Beach and down south in Dana Point and these kind of places, out in Catalina Island, which is beautiful, mind you. But that's all kelp forest area. And kelp is a kind of alga, but it's enormous. It's like diving through a forest. Utterly beautiful. It's one of the most fantastic things you've done. What gets kind of spooky is, there's a lot of beasties that like to live in that. So you never know exactly what's around the bend as you're diving, not just through this forest, mind you, where you really can't see through it. But California waters, of course, are nothing like Floridian waters, especially Caribbean too. So Florida and the Caribbean, you have this beautiful water in some areas. California, not so much. It's really difficult to kind of see through that. And I'll show you a bit of that as we go through this lecture. So again, there's there's a lot going on here. This grape variety, yeah, that's it. I mean, even something like a little pond water that you could pick up. Go and find something in the backyard. Maybe you have a potted plant with some, like a container underneath it that's collected water, and just do that. Sample some of the water there. Go in and see what's there. It's absolutely fascinating. We can go into a pond, get some water out of that, although do be careful, especially here in Florida. Sort of playing around in warm, fresh water, sometimes not the best idea. You might get the Fowlery infection, which, heaven forbid, I hope you don't get that. But it's really one of those scary things. So again, you got to kind of know what you're doing, what you're working with. But even that pond of water can just be fascinating what's there. Seeing all these organisms just flowing about. It's just amazing. And then, of course, trying to identify what's what. That too could be a bit of a challenge. I mean, some of those little beasties just zoom through your field, so it's really quite amazing. You got to kind of slow them down a bit and then sort of take a look at what's going on. It's, again, fascinating. But do note, of course, that Protists are eukaryotes. So when you talk about the prokaryote, again, you're mainly talking about, or you are talking about bacteria, and that's going to be your eubacteria and the archaebacteria. And then with that, everyone else pretty much is eukaryotes. So your protistans, yes, all these groups in here. And then, of course, you have the fungi, the animalia, the plantae, and this is sort of the eukaryote lines in there. We've talked before about what differentiates a prokaryote from a eukaryote, but these guys are definitely eukaryotes. And so, but I've said the classification is tough here. And one of the main issues, of course, is what I noted before. It's not monophyletic, so that presents a unique problem. And as I mentioned again, it is no longer a valid kingdom. So this is getting disbanded out and moved into a lot. I mean, it's really, I can't just say it's moved into a domain. It's just moved into a new kingdom, like we have 3 kingdoms instead of 1 kingdom. It's nothing that easy, unfortunately. So like anything else, you got to look at the morphological issues, the biochemical issues, and of course the genetic issues to try to get an idea of what to do with these guys, and that's proving to be quite a challenge. So as I said, they're eukaryotes, that they definitely are. They have organelles, they have nuclei, the whole works for these guys. And as I mentioned, some can be unicellular, some are colonial. The Volvox, one of my favorites, of course no bearing on anything, but one of my favorites, the Volvox, just beautiful organism. That's a version that's colonial. And some are enormous in size, like some of the algas. It's really just an amazing, amazing group. Because there's so many different types in the kingdom, well, current kingdom, of course, it shouldn't be surprising to you then that they actually use many different feeding strategies that are available to living organisms. Of course, some use light to feed themselves, so these are photosynthetic versions. Algas, for example, will be photosynthetic, that kind of idea. Some euglena might be, they have that beautiful kind of green to them. Some are heterotrophs, so they ingest either organic compounds or the tissues of other organisms. Maybe they hunt a little bit, or they ingest other food particles, phagocytose, maybe another organism, or just particles in the water column. And then the cool ones, not that any aren't cool, but these are really neat, to be able to do both worlds, to photosynthesize and be heterotrophic. How great would that be? Can you imagine if we were like that? Go out in the sun, go to the beach, have a nice warm day at the beach, and feed yourself at the same time. What a nice thing that would be, huh? But these guys can do that, and they are definitely a unique group called the mixotrophs. So along with the feeding types, it should not be surprised as well that there's a whole bunch of different kinds of reproductive strategies out there. Even some can kind of flip back and forth between a sexual and asexual stage. So again, a lot going on here. And we'll take it quickly from group to group as to what's going on here. Now initially, these guys were called mitochondriates. Now remember, in Latin and Greek again, A or AN means without. So these are groups that didn't have mitochondria. Now that might seem a bit surprising because I noted to you before that eukaryotes typically have then either mitochondria or the remnants of mitochondria. So this might be a little bit surprising to see that, but we don't really think that now. We really think that a lot of these guys have variants of this, of the mitochondrial line. So we had to redo the issue here. We had to relook at the group. But again, that's not surprising. It's so unwieldy again, so much going on, that we had to sort of reclassify as we went, and this is no surprise. And here's kind of what I'm hinting at. Now we have some new groupings coming up. It's not just enough to say, okay, we have the classic kingdom, phylum, class, order, family, genus, species. Now we have other divisions we have to talk about. One are these supergroups. So we have supergroups, we have superphylum, we have divisions alone, that kind of idea. There's a lot of issues going on here. For the supergroup, it's one that I haven't really talked about. It's still within the taxonomic scalings that we have, the kingdom, phylum, class, order, family, genus, species. But this is something that's above the kingdom level, but below the domain. That's a supergroup. So again, it's just so many different organisms that might be there. It's got to be bumped up. Not quite to the domain level again, but it is bumped up. And you can see we have a couple lines that are in there. The funny word for us, the Opisthecanta line, Opisthecons. And we're actually within these lineages. Animals are part of the Opisthecanta line as well. And there are others like us. There's the molds and the amoebozoans that could be running through this group. And I noted to you, Campbell's noting 5, but I noticed too that some actually note that there might be 6 supergroups now that might be running through what was the kingdom Protistan line. So again, that one kingdom alone gone, and now bumped up to the supergroup, not quite domain again, but supergroup, and there could be 5 supergroups just within this main lineage. So it's very challenging again to get this squared away. Okay, now with this, if you would please take a look at your book. It might be a little bit hard to see, I do apologize, but if you take a look at your book, you get a nice snapshot of what we think is going on here. Now, this is the main classification schemings that are there. You can see how the phylogenetic tree kind of breaks down from the original ancestor that was there, this grouping over here, down to your groupings, your taxa and your sister taxa, these kind of ideas. But the general classifications are tough. Again, no longer one kingdom. So to give you an idea, and you do not need to remember this for your test, because again, this changes somewhat rapidly. So just to give you an idea, the first one you can see up there at the top, it's called excavata. And that's now actually the kingdom excavata. Some would say kingdom, but others actually think that it's unranked. So that's kind of an odd one. What do we do with that? An unranked area. Okay, so I'll stop. But taking that it's a kingdom excavata, within that, the first one there, the diplonomads, those are actually orders now. So it's not just kingdom, phylum, class. It's all the way down towards the order level where you find that one. Then you have the class underneath it, that's parabasalids. That's actually the class parabasalids. So the order diplonomads, and then class parabasalids, and then the phylum Euglenozoa. So wow, I mean, it's not clean to say, okay, it's kingdom excavata, and then phylum diplonomads. It doesn't work that way. And then maybe under phyla you have classes and orders. Nope, nope, nope. It's not so easy. So again, it's order diplonomads, the class parabasalids, and the phylum Euglenozoa. That's currently how that particular kingdom sits. Again, assuming the fact that you think the excavata is actually a kingdom. Wow, oh my goodness. Okay, and that's just the first one. The next one, which is in the grouping that's called biconta, actually the next two, the chromoviolatas and the rhizaria, these are in the unranked area called the biconta. You have first the kingdom chromoviolata. And within that kingdom, you have the superphylum alveolata. Okay, so you can see the alveolatas. That's right here. Alveolatas there, and the straminopiles are there. So right off the bat, we have an issue going on. You first have the kingdom issue, that's chromoviolata, and then of course the subphylum alveolata. And within the subphylum alveolata, you have the phyla Psilophora, Dinoflagellata, and Apicoplexa. So wow, I mean that's, again, a little bit difficult to classify, and that's currently how it sits. And then the other one, that's the phylum Straminopiles. So again, where the alveolatas was superphylum, this is just the phylum Straminopiles. As a side note, the Straminopiles are also known as the Heterocontophyta. So it's another name of that phylum group. And it pretty much means that they have flagella. They actually have 2 flagella of unequal length. And then within the Straminopiles, you can see there's the diatoms, the golden algas, the brown algas, and Omycetes. But diatom, golden alga, and brown alga, those are common names. So we have true names to them. And in this case, the first one is the class. It's a class Basileophyca. This is diatoms. Then you have the class Christophyca. That's going to be your golden alga. The class Phaeophyca, that's going to be your brown algas. And then the last one actually says what it is. It's a class Oomycetes. A note on that one, when you see mycetes, that's typically referring to fungus. So they were thinking, okay, originally the Oomycetes was a fungal group, and now they realized, wow, look at that, it's not really a fungal group. It doesn't quite fit into fungus, so it got taken out of the fungus and stuck in the protist, and here it is, currently under the kingdom chromoviolata. Again, a bit on the messy side. The next one is rhizaria. That's still in the unranked bicontas. We think currently that it's the supergroup rhizaria. Some say kingdom, others say supergroup. So right there's a bit of contention going on. And within the supergroup rhizaria, you have several phyla. Phylum Cercozoa, the phylum Foraminiferans, which is Foraminifera specifically, and then the phylum Radiolaria. There's, again, not consensus, at least on the kingdom versus supergroup issue on this one. Underneath that, you have the unranked Archaeoplastida. Not everybody thinks it's unranked, however, and we'll see that later on. Now, within the Archaeoplasta, you have what they call chlorophytes, charophytes, and land plants. Again, those are obviously common names. Land plants, that's obviously a common name. And not quite so easy. First off, in here, most of these are known as divisions. Now, divisions is a bit of a note. The division level, which you may not have seen before, is pretty much the same level as a phylum. It's just we use divisions with fungi and plants. Now right there, that brings up a point. Well, okay, red alga, chlorophyta, and charophyta, and of course land plants. Are we all talking about plants here? Are we talking about fungus? Well, obviously these aren't fungi, but what about being plants? And if they're plants, why aren't they stuck into the kingdom Plantae? So, it's one of those issues of, well, they must have something going on that doesn't quite classify them as plants. So you either have a group that says, well, leave them there because they kind of fit, or put them somewhere else, so they got stuck somewhere else, and here they are. So again, it's a bit of a mess. But they did retain the division name, harkening back a little bit to their being in the, at least related to the Plantae. So we have the division Rhodophyta, and that's going to be a red alga. The division, right underneath that, you can see it there, Chlorophyta, and the division Charophyta. Now underneath that, you see land plants. That's a bit of a tough one. From what I think Campbell's going at there, that's actually the subkingdom Embryophyta. And embryophytes are like true plants, and they actually have embryos. We'll talk about that later. And even there, you can have some classifications. Obviously, things like green algas and charophytes relate to the land plants. And you can see green algas are the charophytes and the chlorophytes. They relate to land plants. So you could kind of group all those together. And if you group together the division of chlorophyta, charophyta, and subkingdom Embryophyta, you get what's known as Viridiplantae. That's another way you can classify. So again, not very easy to do. Where exactly do you stick these guys? They're actually not plants, except of course the land plants. Those are true plants. What do you do with the red algas, the green algas? What do you do with those guys? It's been tough, which kind of hints now again why this ended up not being monophyletic for all of these groups. It's just very difficult to deal with. And then the very last one that you see there is the unranked Unicanta. So first off, again, unranked. And then again, kind of like what you saw up here in the alveolata and straminopiles, you see amoebozoans and the opisthecants. So again, those need some classifications. In the first one, for the amoebozoans, right now it's in the supergroup Amoebozoa. And within the supergroup Amoebozoa, you have first off their slime molds. Slime molds right now is polyphyletic. So you really can't classify those very well. So right there's a problem. Underneath that, you have the subclass Gymamoebas, so all the way down below class level, subclass. And then you have the genus Entamoeba. So my goodness, we're not even talking like order higher. These are subclasses and genus level. Then you have the supergroup Opisthecanta. Those are the opisthecants. And then within that, you have the order Nuclearidia, so nuclearids. Then you have the, of course, kingdom fungi. That's been kind of standard for a while now, kingdom fungi. Class Tranoflagellata, and then with that, the tranoflagellates, those are actually most closely related to animals. Beyond that, when you talk about plants, animal, and fungus, animals, of course, are more closely related to the fungi. But when you want to say what's directly closely related to animals but isn't really an animal, that would be the tranoflagellates. And you can see we shared a common ancestor most recently. And that's a class, of course. And then, of course, the kingdom Animalia. All of those, nuclearids or the Animalias, of course, are your opisthecants. So it's kind of neat to think of us as not only a protist, but also being an opisthecon. It's kind of cool. I don't know. I think it's kind of cool to kind of think of it that way. So amazing, yeah? I mean, it's incredible how complex this is. How many organisms are there? What do you do with them? How do you classify them? And very easily, I'm sure it is to see that this is not monophyletic, and therefore really isn't a valid kingdom, which is why everything is so messy. And of course, as I'm saying, some of us aren't even in agreement as to where things are classified and what to do with them. So just hang on, it might change again, and I would not be surprised if it changes again. And of course, even the unranked issues, what do you do with them? So this is proving to be difficult. Now, for your exam, don't worry about everything I've been talking about here. What is a current taxonomy? As always, I follow what's in the material presented by Campbell. In this case, that's the text we're using. Therefore, the formal notes. I'll, of course, follow that for your exam. I just want you to be aware of, well, why is this a kingdom that's being disbanded? Why is this such a messy kingdom? Why isn't it monophyletic? And I hope by now you can see just how unwieldy the classification is. I just wanted you to have a little bit more up-to-date view on that. All right, so with that all being said, let's go right into the excavata. Excavatas, notice we're doing here with the cytoskeleton. And with that, we have some main groups here. I've already mentioned them generally, the Diplonomads, Parabasalids, and the Euglenozoa. Now, with a lot of these groups, there's a lot more in your textbook than that's here in the notes. That's perfectly fine. Again, this is just a quick view of the notes so we can do our little flipped classroom session. So be sure to read the full PowerPoint. But what I expect you to know is what's in those PowerPoints. If you know that, you should be good for the test. Everything else, of course, as I mentioned to you, is supplemental to the actual exam itself. Now with that, the Parabasalids, you can see with that group, modified mitochondria. So be careful on that. which mitochondrial type are we talking about? I would recommend then you know that for your exam. The mitosomes versus hydrogensomes, those kind of idea. I noticed with the diplomomads, they have that one I just mentioned, the mitosomes. Mitosomes have been challenging for us to sort of classify. It's again one of those difficult things. It's not quite a mitochondrion. First off, we don't even know what they really do. It has an unknown function to them. It's thought, at least from current research, that it's involved in what's known as the iron-sulfur cluster, which is its sort of processing. So not quite in direct lineage to what we think the mitochondria might be doing. But we thought that they come out of the mitochondria. So it's either directly from the mitochondria, or it's actually being argued, some think that it might just be remnants of the mitochondria. So it's not quite sure then what a mitosome is actually doing. And with that, notice it gets its energy from anaerobic pathways. Of course, mitochondrial issues are dealing with aerobic pathways. So when you look at the glycolytic pathway, your intermediate reactions, the Krebs cycle, and of course the electron transport chain and chemiosmotic cycle. The Krebs cycle, electron transport chain, and chemiosmotic cycle, of course, is in the mitochondrial issues, and that's, of course, aerobic, in particular electron transport chain, where you have to have O2 as the final electron acceptor of electron transport chain. So this is a little bit different than that idea, and again, be sure to differentiate that on your exam. Notice they have nuclei, not surprising, we're talking about eukaryotes here, and they have flagella, locomotion issues. Now, I've given you some examples of what they can do. It's kind of important to remember that a lot of infectional types can come out of this, including here in Florida. We've been having some issues lately with some of the amoeba infections. And in fact, just this evening when I was getting ready to come in, I saw a report that was coming from New Orleans. And there they were talking about some children picking up some amoeba infections from water, drinking the water. This wasn't water they were swimming in. It wasn't a lake. It was from a water fountain. So the CDC is currently trying to verify that. So bear in mind, this is not amoeba, of course, but bear in mind, we're still talking about freshwater systems often, and we're talking about infections that can happen, including with you and I. And notice this one is an often a parasite issue, causes what's called beaver fevers in humans. It can be quite severe. I noted to you that people who have compromised immune systems, this can actually be fatal in them. So you got to be very careful with this. Know your particular health, know the environment, of course, you're getting into, what might be dangerous for you in that environment so that you stay safe as always. So it would be a very good idea. And this is one of the infections you can get out of it. There it is. It's kind of a unique looking organism. You see those flagella and it's shaping here. I'm not a big fan of having a picture on your test and asking you to differentiate this particular protistan from another. That's more of a lab issue. But it's kind of intriguing to see what they look like and seeing some of the sizings. And you can see it's only in the micron range in this case. So very small, but still can have an effect on you and I. The parabacillids, in this case, they have those hydrogensomes that I noted to you. This is also dealing with how they produce energy. They do produce out some ATP from this. Notice it's also anaerobic that they're producing out. They make ATP from that, like I just said. They make hydrogen out of it, at least use it. They have acetates and CO2 issues that flow through here as well. But it is an anaerobic process. And once again, I've noted to you an infection that runs through this, the vaginalis infection, that can affect both males and females. Some people think, and I get it from students, oh, it's a vaginitis issue, which means vaginalis. It's only for women, and that's not true. Notice in males it causes urethritis. Bear in mind, something that ends in itis is referring to an infection. So this is urethral infections that might be running through here. So again, you've got to be a little careful about this. And there it is. You can see the flagella here, and these membranes are used for undulation motion, a typical sine wave that's often used in nature for them to move around with. And then again, very small. You can see it's around 5 microns, that issue there. So you can just add up the sizing on it. But again, very small. For your euglenozoans, quite a few here. Notice these can be predatory. So I mentioned to you earlier that some of these protistans are indeed predators. I mean, it's kind of cool to think about that. This little organism you can't really see with the naked eye if at all, swimming around hunting. It's cool what they can do. And in fact, who they can hunt is really quite amazing. Obviously, some are photosynthetic. We saw the photosynthetic version in lab and some of our prepared slides. And they have this crystalline rod that's in the organism. Interesting to see that we yet in the sciences don't know what it does. As I kind of teased before, that's kind of nice. It's a bit of job security. So just one more thing we can go out there and study, which is exciting. There's still a lot to learn in nature, of course, and that's wonderful for us to have. But it's part of their flagellary issues. Now, when you look at that crystalline rod, it looks a bit like this, obviously our artistic rendition. But take a look here. This should look familiar to you, at least in a way. Notice there are rings of 2 here. So you have 1, 2, 3, 4, 5, 6, 7, 8, 9, and then 2 in the center. That's a 9 + 2 microtubule arrangement. And that should be ringing a huge bell to you. We've seen that before. If you remember your biology, so Bio 1 in this case, that would be, of course, the cytoskeletal elements that we saw that in. Be careful, though. We're not talking specifically about our cilia and flagellum. That takes that 9 groups of 3 microtubule arrangement. This is the 9 groups of 2 that you see through the crystalline rod. For the kinetoplastids, one of your other groups that are found in there, they have the kinetoplast. That's their main structure there. Notice they can be found almost everywhere, and they also cause infections in us. This one is the sleeping sickness that's found in us. One that's not noted in your textbook, but I wanted to note is Chagas disease. It doesn't come up very much, but it is out there. And I've noted to you that it can affect several organisms and lead to cardiac problems if you don't take care of it. And those are indeed life-threatening again. So you do need to be careful with these guys. Here you can see that protist here. Absolutely beautiful imagery here. The microscopy is amazing. You can see those organisms here, and of course red blood cells around them. It's really amazing. Again, the sizing is just so small, and it's kind of amazing to think that it can cause us so much problems. For the euglenids, you can see these guys are quite small as well. At least they are. Some are autotrophic and heterotrophic, and we've seen some of these. There you go, a photosynthetic euglenid. There's this eye spot, a flagellum. I noted to you in lab that you can't really see the flagellum well under high power. It's a little bit easier to see it under oil immersion, but you can definitely see it. And there it is, a beautiful little organism. For the chromoviolata, another kingdom for you in this case. So in this one, notice it is monophyletic. We're trying to work out all the classifications still with these guys, and it was thought that they had an endosymbiont issue with red alga. Through this, we have the violata group that I talked about a few moments ago, and the straminophiles. So here's your alveolata. In this case, they have these alveoli. Now be careful, these are not the alveoli you might talk about in our pulmonary system, but they do have alveoli. It wasn't quite sure what the alveoli do. There you go, not quite sure what they do, but there is some indication the alveoli are used as an armoring system. Same with like the dinoflagellata. There you go, the dinoflagellata might use that in that armor that's found in the organism. So with that, you can see the main groups. I just mentioned dinoflagellata. The apixoplexans, they have a structure called an apex. Apex does mean like top area, but it does have function for them. And then the ciliates that are found here as well. There's that alveoli. Again, was thought to be a bit of an armoring system on them, even though we're not entirely sure what it does. All right, so dinoflagellata, very well known. Not so much in regards to we've described them all, we have a perfect understanding, I don't know about that, but it's more of we know that these have big effect. We see them because we see them in blooms, algal blooms. Of course, the most famous, of course, is the red tide blooms. That, of course, happens naturally. You just have these right conditions that occur, and you get this huge like boom of organism. We call it a boom-bust cycle. Big flourish of them. That's the algal bloom. The downside of that is, as the algal bloom happens, the organism itself, the dinoflagellate itself, can be toxic to some organisms, like the fish. And then on top of that, they tend to strip out the water column, making it hypoxic. And on both those, organisms begin to suffer and die. So it could be quite a problem with this group. It was thought for quite a bit, too, that, well, you know, that's more of an aquatic issue. Yeah, it might affect marine life, for example, but it's not really much harm to humans. So for a long time, we said, oh, yeah, don't worry about it. Go swimming, have fun. Yeah, the water looks like it's blood, but hey, it's not blood, so knock yourself out. And that really isn't the case anymore. We do think that the red tide might actually be hurtful to humans, especially if you suffer from things like asthma. So we do recommend that you avoid messing with the water column while there's having an algal bloom outbreak, a red tide outbreak. So do be careful with that. Excuse me. Notice that they are diverse. They have a lot of different feeding mechanisms. And of course, here's your red tide outbreak issue that I was talking about. There you go. It doesn't look like much. Again, very small, but that is a kind of this organism. Now with that, I wanted to show you some of these things. It's again a little bit scary how this works. So to give you an idea first off of how the red tide outbreaks, and then I want to kind of show you sort of what it looks like.
From a red tide algae blooming off the coast of southwest Florida. Red tide algal blooms release neurotoxins that can kill marine life. They can also lead to oxygen depletion in the water column, resulting in hypoxia. Marine animals that consume contaminated fish can also be harmed. Neurotoxins can also become airborne and drift ashore, causing coughing, itchy eyes, and respiratory problems in humans.
Okay, so that's kind of an idea of how it works. Now what it looks like, again, it can be absolutely striking. Here's one that was happening in Australia, and you can already see some of the tinting red here and some tinting red in here as well as some of the algae are blooming and moving with the currents.
Bondi dyed red. 4 kilometres of Sydney's coastline transformed by an algal bloom.
It's not something that's going to hurt anyone. You might come up with an irritation on the skin or eyes and things like that, so that's why we took precaution to shut the beach down.
The quiet waters of nearby Clovelly were also hit by the red tide, a spectacle of nature no one here has seen.
Yeah, I haven't seen anything like this down here before. See a lot of seaweed and out and sort of algae at times, but nothing like what it is today.
The thick algae starved the water of oxygen, killing fish.
I'm not that brave. I like to keep a distance from things that look red and scary.
Lifeguards warned swimmers to keep out. Not everyone listened.
We try and get in the water every day, so we're not going to let a bit of algae deter us.
Experts spent today testing water samples. They showed this bloom was caused by Sydney's recent hot weather and heavier rainfall.
The water temperatures come up from 18 to 21 really quick, and with the outside heat as well, it's a combination there.
The surf at Bondi is expected to be cleared by tomorrow, but it could take longer for the tide to wash it out at more protected beaches.
Okay, so you might think again, well, you know, that's in Australia, doesn't really happen perhaps in some other areas, but of course it does. So here's one not too far from home. Now, as I noted to you, these can happen for natural reasons, but they can also happen for artificial reasons. So, for example, if we have an area where people are putting a lot of nutrient into the water column, and that can be everything from potential detergents you might use, although those phosphate-free detergents aren't very common anymore, all the way down to you're putting too much nutrient on your lawn. You want this nice, like St. Augustine, whatever grass it is, to really be growing well all year round. So you're putting down massive quantities of nutrient, a lot of nitrogen and phosphorus, even iron. Iron could be a limiting compound, element that's out there. So it's a limiting nutrient in the water column, and that could be a bit of a problem. It runs, when things run from your golf course again, or your lawn, or whatever it may be, it runs to the lowest point, and that lowest point is eventually going to be the ocean, and then it spikes the dinoflagellates, so the algae spikes, and you get the hypoxic events as well as neurotoxicity, and there is going to be problems. So it's unfortunate, but these things do happen. For your picoplexans, as I mentioned to you, the big note here is the apex, the structure here. That apex is thought to help the organism sort of penetrate into hosts. However, we're not exactly sure how it does it, but there is some indication it might be from other functions like lipid synthesis that might be going on here as well. So there's been some push to target that and to be able to look at ways of affecting the lipid synthesis, kind of blocking lipid synthesis. And if you can do that, you might block the effect of the apex and therefore block its effect of infecting a host. Notice that when it does, it creates these things called sporozoites, and that creates quite a bit of problem. They have both sexual and asexual stage, so they need hosts to complete this. Now, when you look at host types, this is a very specific one. First off, notice this is capitalized and italicized, so we're noting a genus here. And the Plasmodium is the Protistan that we think is actually affecting humans, causing malaria. And a lot of people are affected by malaria every year. So there's a push to try and stop this. Even some scary pushes, like going back and starting to use DDT in some places, which is pretty much like banned across the planet. But the catch here as well, we know DDT is maybe very harmful to the environment, but now we have a big problem with mosquitoes. There you go, mosquitoes, and with that, this problem of malaria. So it's kind of this balancing act you're trying to do. But we're trying to do things to get in the way of this organism's effect and therefore causing harm to you and I. So when you look at its life cycle, if you look very closely, you can see a mosquito right there. And the mosquito then is affecting the human, it's biting them, and the little protistans going in. Notice it has effect on the hepatic system, so it affects the liver. And when it does, there's the structure being formed, the merozoite is being formed. Once it's created out of the hepatocytes, it goes into the blood system, gets into erythrocytes, and the erythrocytes creates the gametes. The gametes are then picked up by another mosquito. You can see that little mosquito right here. Once it's picked up by another mosquito, the gametes are going to fuse, they fertilize, create the zygote. The zygote then goes and creates more of these sporozoites, and they get picked up by the human again, another human perhaps by being bit by a mosquito. So you're trying to disrupt that system. And with that, this is where compounds like DDT has made its resurgence, because you're trying to kill off the mosquito. And if you kill off the mosquito, you break the cycle, and therefore malaria hopefully doesn't continue to have high rates of it. Very scary, though, to think of countries going back and using DDT, but they had the reason in this case dealing with malaria. Not that I'm justifying that in any way, but it is intriguing to see how we're trying to approach the problem. For your ciliates, it is correct. There's a lot of them out there, and kind of nice. We call them ciliates because they use things like cilia. So what do you know? It actually makes sense. Now here, they have a very particular reproductive cycle, and they go back and forth by use of this macro and micro nucleus. So you need to focus in on how is it reproducing and what are they doing to replicate out the micro and macro nuclei. They do it in their version of sexuality. Notice here, though, the conjugation is a sexual process, but in this case, it's different than reproduction. That's actually this division that happens. It's like a cytokinesis issue that happens so that they can, through binary fusion, create daughter cells. All right, now with that, I would recommend then for your exam that you are familiar with the reproductive cycle of something like this. This is your paramecium. So if you take a look at this, you can see two paramecium that are of correct mating types that come together. They have this partial fusion, and in that case, now they can start to work with their micronuclei. They actually replicate out the micronuclei. In this case, be careful, how they replicate it is important. Notice it's a meiosis cycle. See, is that right up there, the meiosis that's happening here. And after meiosis is complete, they've created their micronuclei. Most of micronuclei disintegrate, and they're left with two in each of the paramecium. Once that occurs, they then swap micronuclei, so you can see each end up with one blue and one purple micronucleus, and then they separate. They're pretty much done with their version of sexual reproduction. You know, they've gone through their sexual stage, and they've swapped genetic material. Pretty much that's it. They're done with that. Now that you have the paramecium with its micronuclei, the micronuclei then fuse. So these purple and blue micronuclei fuse into a single micronucleus. That single micronucleus then goes through several rounds of mitosis. So be careful down here. They're noting to you, and mitosis creates out these 8 micronuclei. So mitosis here, meiosis up there. Make sure you differentiate that on your exam. Once they have created the 8 micronuclei, several of these micronuclei are sort of grown into macronuclei. The other 4 are made micronuclei. They then go through cytokinesis along with binary fission to create out 4 daughter cells. Each daughter cell has its own micro and macro nucleus. Then they go off on their way. So this was the reproductive part. They go off on their way. They're done with the whole process. Make sure, again, that you can differentiate the steps here, including how the micro and macronuclei are formed for your exam. All right, for the straminopiles. In this case, as I noted to you, we call them this because they have 2 flagella of unequal size. And it's kind of cool. They have one hairy flagellum and one smooth flagellum. And with that, we have quite a few groups. The bacillio groups, the golden algae, brown algae, and of course the one I mentioned to you that's kind of like a fungus but not really a fungus. So there you go, you can kind of see that. One is hairy, one smooth. There you go. So make sure that's clear enough for your exam. And then with that, the first ones, your diatoms. Now diatoms, fascinating, absolutely fascinating. They're small again, but they make up a large part of what's called the phytoplankton. Phyto referring to light, plankton are organisms that really can't fight too well against the currents. They kind of go with the current. Now the ocean itself, the primary productivity of the ocean is not very high if you look at it just in one little area. But because the ocean is so massive, overall, it becomes an important part of the primary productive system of the Earth. So unlike, say, a part of a rainforest area where a small area can have huge primary production, but then again, we don't have a lot of rainforest necessarily on the planet, especially the way humans are kind of ripping it out. The ocean's a bit reverse of that. Not a large primary production, but there's just so massive a system that it becomes important. And the diatoms are an important component of that. So with these guys, one of the more interesting things, of course, is that they have what's called hydrated silica that they use pretty much as their glass system. So they have a body made out of glass pretty much. There you go. Their body made out of glass pretty much. And it's amazing to think not only that we have organisms made out of glass, which is very exciting for us as scientists, especially if you're a sci-fi fan, you're like, oh, look at that, it's a silica-based life form, you know, that kind of issue. But their body is, of course, made out of glass, and they're not the only ones. But then also with that they're photosynthesizing, some actually ingest organic compounds. You can see that they reproduce, a lot of them do so asexually. I mean, it's just an amazing, amazing group, all of which is doing this in a glass-like body. Just fascinating to think that an organism can do that. They leave a good fossil record, and then they create like this powdery material that's found in the rock. Of course, it can kind of do its version of lithification, and that's known as diatomaceous earth. This could be used for a lot of things. When I worked in aquariums, we'd actually take bags of diatomaceous earth and use it as a filtration system. So we'd have water that would percolate over the diatomaceous earth, and it was very good at pulling out particles, helping to clean up the water column. So this actually has a lot of use for us. And then with that, you can actually see some of their sizing. I mean, it's just beautiful. Look at all the different shapes they can form out of glass again, of all things. Just absolutely phenomenal. What you can see here, and then here's an SEM version of that. But they can take a lot of different shapes to them, you know, being able to manipulate this glass-like structure. Here's one of the most famous aquariums in the world, the Monterey Bay Aquarium, another view of diatomia, showing that it's got this very ornate manipulation of the hydrated silica. Another version here, some people might think, oh, that's a sea star, but it's not a sea star. This is actually just a shape that a diatom actually took. I mean, again, absolutely phenomenal what a glass-like organism can actually do. For the golden alga again, in regards to golden alga, these guys are of course, Christophyca that are out there. They're not very large, and they get that kind of that coloration to them because of the carotenoids that they actually have in their tissuing system. Notice they can be flagellated, and a lot of them are photosynthetic, along with some being mixotrophic. I mean, pretty straightforward group. Here's a colonial version that you can see. And it doesn't look quite golden in this picture, but real ones actually have a bit, in my opinion at least, a bit of a golden tint to them. For the brown alga, these guys are the largest of what we call the seaweeds. Now, there you go, seaweeds. Now be careful, seaweeds, that's a very general term. It's like saying plant to me, although remember, these are not plants. It's like saying plant to me. It doesn't mean very much. It's not specific enough. Look at it that way. If you say plant to me, okay, what kind of plant are we talking about? Loosely, any gymnosperm or angiosperm can be called a plant. So which one are we talking about? It doesn't have to be a pine tree. That's a plant. Okay, and doesn't even tree, that kind of idea. If you say tree to me, I don't know what you're talking about. Is it gymnosperm, angiosperm? Plant? Is it gymnosperm, angiosperm? Is it a bryophyta? Is it a pterophyta? I mean, it's a very general term. So here, if we say alga, first off, or not alga, a seaweed, remember, first off, we're not talking plant. And then what seaweed are we talking about? Is it the red alga that could be a seaweed? Is it a brown alga that could be a seaweed? You've got to be a little more specific than that. But these guys are definitely complex. They're not plants, so we don't see the version of vascular tissue. We don't see a xylem and phloem here. We don't have roots here. They have what's called a holdfast, but they're not roots. So again, they have quite a bit of difference to them, which is one reason why it's hard to classify them as a plant. So some of them in anatomy have the thallus. The thallus is actually the vegetative part of the plant. This holdfast, as I noted to you, is like their roots, but bear in mind this is not roots. It just helps to hold everything down, so it's a good name for it. The stipe is like the body, and then blades are blades, and that's kind of similar to what we talk about, say, with a plant. So there you go. When I was living in California, especially as I went up north a little bit, I saw this everywhere. You can see the holdfast there, that stipe. By the way, that's stipe, not stripe. Again, be careful on test. Stipe, and then, of course, the blade system on top. Absolutely beautiful. And this is what I was referencing at the beginning when you could actually be diving in like kelp forest. Incredible, incredible thing to do. I highly recommend it if you get a chance to ever do so. Reproductive, again, as I've mentioned to you before in class, be careful. The keys here, this like off like red color is a little bit of the it looks red to me, but it's the diploid system, and then of course the off blue is the haploid. You can see here as it matures out into the blades, you have the sporangia, so spore-like structures. The spores are released. Once they're released, they create what's called the gametophytes. Now in the gametophytes, you have two structures we often talk about, the archegonium and the antheridium. If you're talking archegonium, that's referencing the female part. If you're saying antheridium, that's referencing the male part. So in the males and their antheridial kind of structure, they're going to release a sperm. They already have a water medium. We're talking about, say, a brown alga, so like a kelp and marine system. So there's the ocean right in front of you. And then, of course, it moves around. They might be attracted over to the egg. They fertilize the egg, and now you're on your way to your zygote. You have that fertilized egg, develops another spore site, and you're on your way, of course, to another kelp system. Pretty direct. Now, just to give you an idea, it's just phenomenal being able to see what kelp forests are like. And they're very important. This is, again, like a forest under the ocean. So with that, productivity issues come up here. Like a forest, you have a canopy, you have a midsection, you have, of course, where an understory would be, so that's down near the bottom. But you have these zones, obviously, and organisms hunt in the zones. Some use them from nursery grounds, some feeding grounds. It's just a very rich, dynamic environment. I wanted to give you a little bit of a view what that looks like.
Can be seen along much of the west coast of North America. Kelp are actually large brown algae that live in cool, relatively shallow waters close to the shore. They grow in dense groupings, much like a forest on land. These underwater towers of kelp provide food and shelter for thousands of fish, invertebrate, and marine mammal species. Kelp are very simple organisms that consist of a holdfast, a stipe, and blades. At the bottom is a root-like structure called a holdfast that anchors kelp to rocks and other materials on the ocean floor. Young kelp must compete for space to settle and grow, as the rocky bottom is carpeted with smaller algae and invertebrates like anemones and sponges. The stipe is similar to a plant stem. It is strong, yet flexible, allowing kelp to slide in the currents of the ocean. Many fish use this middle area of the kelp forest as hunting grounds. The blades contain a special gas which act like a float, keeping the kelp blades close to the surface of the water where they absorb energy from the sun. This ability to float raises the kelp to the sea surface, forming a dense canopy. The canopy serves as a nursery or brood area for many organisms due to the warmer surface temperatures and slower water currents. Many organisms, from small fish to birds and even whales, use the thick blades as a safe shelter for their young from predators or even rough storms. Kelp forests have a greater variety and higher diversity of plants and animals than almost any other ocean community. NOAA scientists study kelp forests by visiting the same locations over and over to assess the presence and abundance of a variety of organisms. Monitoring allows marine scientists to determine if the kelp forest is changing over time and to identify the cause of those changes, whether natural or human. Healthy kelp forests maintain the existence of thousands of plants and animals, fish stocks, and many ocean and tourism-based businesses. All of these require a thriving ocean ecosystem. We all depend on the ocean for food, oxygen, and even life-saving pharmaceuticals. Do your part to keep our ocean and local waterways clean, litter-free, and healthy.
Absolutely incredible. And if you get a chance to go diving in this, I highly recommend it. The Oomycetes, again, if you see mycete, that part of a word, it's referring to fungus. So these were initially thought to be fungus, and we figured out, well, it's not quite fungus-like, so we put it somewhere else. And of course, we didn't know what to do with it, so here it is in our junk kingdom, pretty much. So there it is. They were thought to be fungus at first, and now they're not. They have these things called hyphae. The hyphae pretty much are, for fungus at least, again, it's like vegetative part. It's a part that grows, and here it helps with nutrient uptake. Again, not too unlike a fungus. And the ecological issues can be extensive, especially when you start talking about true fungi. Fungi themselves, not so much here necessarily, but fungi themselves can be absolutely devastating towards plants. Animals kind of get a break here. And we still obviously have fungal infections, and when we do get them, they can be, again, absolutely devastating. But not quite to the degree, say, that maybe plants do. So we caught a break. In this case, you can see it's affecting potatoes that are causing a bit of an issue. For the reproductive scheme, you can see up here they have an asexual version, creating that hyphal mat that they can go through. When the condition's right, it can default into the sexual. They have an oogonium. Now, that's dealing with something like an archegonium. You have the antheridium and the archegonial structure. But you can see here the oogonium has the eggs. The antheridial structure will be kind of like where the sperm is deposited. So sperms are deposited in, you create the oospores. The oospores are released, they germinate out, and now you can either go into the asexual stage again or go back into the sexual stage. It's kind of dependent on what's available to the system. A very direct reproductive scheme. For the rhizarians, the grouping rhizaria, again, it's kind of hard to deal exactly with classification here. Rhizaria right now is in the supergroup Rhizaria, but again, some don't agree with that. Some think it's actually the kingdom Rhizaria, but at least it is monophyletic, so that's kind of nice. We have the classic amoeba that's in here. Again, a lot of press on amoebas lately. I've talked about the pseudopodia before, being able to stick out these false feet. It's by use of the sols, the cytoplasm. They can push into these areas, extend out the cell membrane by the pushing of the cytoplasm, and then be able to extend out those feet. So they can slink around, they can ingest things, that kind of idea. And you can see there's some main groups here, sticking out those pseudopodia, the radiolorans. One of the most famous, Foraminifera, it's kind of a funky word, so they do call them forams. I'm not sure if that's in your text, but I wanted you to know the common name. And cercozoans. Not much to say with cercozoans, just a small little photosynthetic group that's out there. With the radiolorans, notice in this case, another organism whose test is made out of silica. So it's not just the diatoms that are actually having a body made out of glass. Here's another one. And these have these like long cytoplasmic projections, so that's a long pseudopodia. They stick out, and they use that to phagocytose other organisms. And you can kind of see that here, what one of the organisms looks like. You see those long pseudopodia that can stick out. For the foraminiferans, a lot of interesting group here. Instead of having the tests made out of silica, these guys have tests made out of calcium carbonate. So in that case, it's the chemical issue of sedimentary material that we're dealing with. Here, though, they also have these little pores in the calcium carbonate shell that they have where they extend out the pseudopodia and they bring in food that way. But these are very important in regards to classifications. So in the geological calendar, you can go back and look at sediments and see in the sediments, well, what kind of foraminiferans did you have, and therefore what's happened over time in this environment, in the marine environment. In Florida, we've done that. I've been involved in research groups that have done that. They go out in areas, they collect sediments and bring sediments back to lab. They sift through the sediments, actually looking for the foraminiferans. And when they find them, if they find them, they're able to chronicle some of the geological history of the area. So they do become important in the fossil record. And with that, you can see kind of what they look like. Here's that calcium carbonate shell and those extensions of pseudopodia. So with that, I want to give you a little bit of an idea, just a little bit more of an idea of the foraminiferans. Some of the work I've done was pretty much this. You have a sample of sediment you're working with. So it could be a little more gravelly like this, or just true like beach sediments, so silica material that you're working with. And remember, in beach, you have a lot of quartz and a lot of feldspars. So you're kind of sifting through this. Literally, if it's small enough, like if you just have quartz and feldspar, a classic beachy material, as you're sifting through that under a microscope, you have this just a little forcep, and you're pushing gravel, little pieces of beach out of the way, pretty much, a little feldspar this way, a little quartz that way. And in and amongst that, you're looking for the forams, and they're really that small of an organism. So you might come across some. Here's a few that you actually find. You can see easily some of them here. Obviously, these have classic shapings to them. There's another one here, for example. You can see some glass material here, so you're pushing some of the quartz out of the way. Another view of them, this is some of the SEM view of them. You can see they take very definitive shapes. Of course, if you don't know what you're looking for, you might go right by that thinking, oh, that's just a piece of beach, nothing to be too concerned about. But they have a lot of shapings here. And then when you find them, it's really very taxing work. You're sitting there under a dissection microscope, pushing all of this quartz and feldspar out of the way. You actually find a foram, and then you kind of have a little brush with some water, you kind of spear it, and then you move it over to a microscope slide, and you keep doing that for everything you find. Then you take that microscope slide, put it under a compound light microscope, and then try to classify it. And you're trying to get it down to the genus or species if you can, to find out exactly what it is. And then based on that, you get counts, and based on counts, you get a general idea of biodiversity and abundance that's there, and start to build up your paleo environment. Very taxing work. But here in Florida, it's very useful, again, to sort of classify some of the sediments that are out there. Oddly enough, though, one of the sediment types I was working with was devoid of forams, and that was very surprising to everybody. I mean, it's neat when you find them, that's very, very interesting, but to say, well, there's none here. So then you ask the question, well, why? What happened? Why is there none there? Why do you have them a lot in Florida, but in this place you had none of them? So it's an intriguing question. And it's a surprising question, but it's been an intriguing one. What happened in that environment? Why are they devoid? And what that might mean for other organisms. This is a very important view in regards to just how proliferate forams and organisms like them were. These are the White Cliffs of Dover. And for scale, you can see humans here in the foreground, some humans in the background. There's a lighthouse for you there. Not a lighthouse, but it's like a towering system there for lights and that kind of idea. And this area, which is found in the southern part of England, is pretty much made out of small organisms like forams. I mean, there's millions and millions and millions of them that come together to make this material. So you might think, oh yeah, that's igneous rock, but it's not igneous rock. It's not really a mountain. This is really just a lot of dead bodies, pretty much, making up this side of a mountain. So it's really quite amazing when you start talking about something microscopic that I'm sitting there under a dissection scope looking for with little tweezers. I mean, that's how small these guys are. To start talking about how many we actually needed to actually make the White Cliffs of Dover, it's just mind-boggling. A very fascinating thought. All right, so enough of them. Moving on to some other alga types. You have the red algas and the green algas, of course. With these guys, they're photosynthetic, so we're talking about endosymbiont events as well. And you can see that here, that they have some issues of bringing in some other organisms, in this case cyanobacterial issues, and being able to use them for photosynthetic process. This also becomes useful later on, especially in the green alga and the cyanobacteria. When you start talking about things like lichens, lichens are associations between a fungus and either a cyanobacteria or a green alga. So it's kind of intriguing, again, not only in the lichen groups, so you start talking about fungal groups, but also here that we see these symbiotic issues cropping up to form things like the red algas and the green algas. Here you go, the red algas and the green algas. Now notice here, Campbell is referring to the Archaeoplasta as a supergroup, but as I noted to you earlier, it may not actually be a supergroup, that the Archaeoplasta might actually be unranked right now. So again, there's these discussions still going on. As always for your exam, I'll follow this. All right, so here's your red alga. In the red alga line, remember we're still talking about divisions here that are going to be in the red alga, and this is of course the division Rhodophyta. And in that case, they get that deep red pigment from this guy, the phycoerythrin, that's actually in the organism, kind of masking out some of the chlorophylls that are there. And you can see they take some various colors to them. But with this, they could be pretty large. We still call them alga, and people are known to try and ingest these. You can see some of them here. So down here, for example, you can see some humans that are drying it out, and we call that nori, which you might have heard of before. With the green alga, in this case, it's another divisional group again that we're talking about. So remember, divisions are very much like phyla, but just used for things like plants and fungus usually, but here for a Protista line. And for the green alga, we're of course talking about the chlorophytas. So with that, you have the chlorophytes and the charophycans, so they are the charophycans, and these gave rise eventually to the charophytes to land plants, the embryophyta that are out there as well. But unfortunately, it is paraphyletic, so again, the classification is not nearly that easy. A lot of diversity here. You can see some are colonies, some have these large bodies to them. You can see multicellular bodies. This is one a large body, Caulerpa. Caulerpa is fascinating. It's actually one large cell that's making up the entire organism, and that cell has a lot of nuclei, so it's multinucleated that runs through. But it is still a single cell for the entire organism. And then the Volvox is your colonial version that can be found. So here's a couple of views on them. This is ova. Notice it's called sea lettuce. When I was living in Hawaii, I actually knew people that would eat it like lettuce. So they would be walking along the beach, they would pull up some ova that might be blown in on the beach, they would maybe grab some mussel that was growing on a rock, and there you go, they had lunch. I never tried myself, I don't know why I never did, but I never tried it myself, but I hear it's pretty good. It's kind of crunchy and tastes like, well, salt water. So it really wasn't terrible if you like that kind of thing. There's Caulerpa. I mean, again, it looks like a plant, for example, but again, it's not a plant. And B, this is actually one organism that's one-celled, making this entire structure here. It's just a multinucleated single cell for the entire thing. Every leaf, every blade, it's all still pretty much one gigantic cell. Absolutely amazing what organisms have devised out there. Pretty cool. And then, of course, the Volvox being this colonial system. You can see the structuring here. These are the daughter cells that run through. Again, a beautiful colonial organism that's found for the green alga. In the reproductive scheme, again, pretty direct. You can see the zoospores here. When they go through the asexual, they can go through the process of creating more zoospores. When the environment's correct, they can go in their sexual version. Here are different mating types. If you look closely, you can see the plus minus here. They come together, fertilize, and you get the zygote. The zygote opens up. You have more mating types. They continue either into the sexual or move back into the asexual. Again, a very direct reproductive scheme. And lastly for us, the unicons. So that's going to include you and I. And notice, like I said at the very beginning of this whole discussion, the amoebozoa and the piscicons are found here, and the piscicons, of course, include the Animalia. So it's, again, intriguing, depends on point of view, to talk about not only you and I being one of the unicons, but also being an piscicon. That's kind of cool that we can do that. And you can see that we really don't know the taxonomy yet, like I was talking earlier. Maybe one day we'll figure it out. So here's the amoebozoans. They're going to have the gym amoebas and the entamoebas. Again, classification is tough here. I notice these also, like the amoebas I talked about earlier, use pseudopodia. Now with that, the one group that's really tough to work with are these guys, the slime molds. These are polyphyletic, so again, there's no clear taxonomy here at all, but they're just darn cool. These guys are either taking a plasmodial shape or they're taking a cellular shape. And the plasmodial shape, which is known as the plasmodial slime mold, this one here, first off, careful on name, Plasmodium. This is called Plasmodium slime mold. Don't confuse it with the Plasmodium I talked about earlier. Notice the one I mentioned earlier, which I reiterated here, capitalized and italicized, so that's meaning that the genus here. This is not. This is just a Plasmodial slime mold, a little bit different idea. This is very much like a big ball of snot, pretty much. And in the right time, it's aggregated together. It works like a feeding body. It slinks around like on a rock, finds something that it wants to ingest, it wraps around it, phagocytizes it, and pretty much digests out the material. It's really amazing to think that here's this ball of goo, pretty much, slinking around places and digesting organic matter. It's really, again, just very cool. And if you're a fan of the old B movies, well, there you go. Here's a 1960s Attack of the Killer Blob running around, although it's not going to get you, don't worry. So again, very cool. And you can see it's multinucleated, it has, or diploid nuclei running through, and as I said, uses the pseudopodia for phagocytosis. So here's kind of what it looks like. You can see it can go back and forth between the flagellated cell and pretty much a classic more amoeboid cell, depends on the environment. Again, it finds itself in, creates that feeding body I just mentioned, which can kind of slink around on rocks. There's a mature version. You can see it's kind of greenish in some are yellowish in others. Look at it under a slide. They do cross-section of this. It just looks like a smear on a slide. It makes sense. It really is like a slippery ball of goo, pretty much. That's pretty much slinking on these rocks. When it wants to create more spores, it creates a fruiting body that can grow out of this a mature plasmodium. There it is. A mature structure creates the spores, which gives rise to the amoebas again. Really quite cool. And of course, there's no way I'm not going to show you that one. It's just too darn neat. So here is the plasmodium that's just decimating a piece of pita bread, of all things. Now, of course, bear in mind, this is sped up, so this is time-lapse. But down here, you can already see that this thing is just oozing and pulsating. Well, I reference it as a ball of snot. I mean, that's not the most technical, I suppose. But it really is what it looks like. It's really just fascinating. And you can see how this would be a great inspiration for those 1960s Attack of the Killer Blob. It's just very cool to see this happen. And there's that pita bread. It stands no chance with this thing coming at it. Just fascinating. There you go. I mean, that's just so cool to watch. Absolutely amazing. Now, again, this is time-lapse, so this is why I was kind of teasing about the old B movies. You probably have plenty of time to run away from this, but so I wouldn't worry about that in any way. But still, like I said, that pita bread had no chance in this regard. All right, so that's a plasmodial slime mold. Here is the cellular slime mold. These are more true to like amoeba types. Some actually think they have this version of like intelligence in a way. They kind of have their own like to do. They do what they do, and then when the environment's right, the cues are right, they kind of pull together as a single group. So that means you have all these individuals that are functioning pretty much as a single organism. Then from that, they have a fruiting body that sprouts out. Some of the amoebas are going to function pretty much like a stalking system. On the top of the stalking system, you get a fruiting body that opens up, releases spores, and you move on to the next generation. And by the way, the ones that work at the stalking body are going to die in this process. So again, it's self-sacrifice along with what some people consider to be a bit of like intelligence. So it's an amazing aggregate that's coming out of these individual bodies. And you can see that here. Here's the emerging amoeba. And again, they can go through their sexual and asexual forms. In the sexual form, they're going to create new amoebas that are sexual in that regard. Or they can create this fruiting body. And in that case, all those amoebas are kind of coming together. This fruiting body is growing out of it. And you get some that make up the stalk. You can see that here, and some that are making up the actual fruiting body where the spores are. And at least the ones in the stalk are going to die through this process. So it's not only kind of intelligence that people kind of talk about, although sort of put that in quotes, but it's also self-sacrifice. And we do have a video on that. This is a gentleman that actually has studied them for most of his life, trying to highlight what they do and how they do it. Again, pretty darn neat. Okay, for your gym amoebas, you can see these guys are found in freshwater and marine environments, and it's a classic like amoeba again. The amoeba Proteus is actually the one we had in the prepared slides. In lab, you could take that cytoplasm again, that solid material that I was referring to, they kind of push it up against the plasma membrane and then get these pseudopodia to kind of push out. As I was kind of teasing in lab, the defining characteristic of these guys is that they really have no defining characteristic. They could take almost any shape they want. This is a very malleable system. And you can see they kind of hunt around. They're heterotrophic and they go and attack other things and kind of bring them in. With the entamoebas, these guys can definitely be parasites. They can go after organisms like vertebrates and invertebrates. So you got to be kind of careful here. And they're noting to one of the infectional types, in this case dysentery, amoebic dysentery, that humans can pick up. So again, these guys are found everywhere. Ecologically, they do a lot. I mean, they're hunters, they're photosynthesizers, they make up a lot of the phytoplankton, so they're very important to the food web system. And bear in mind, the primary producers are critically important. From that, you get the primary consumers and the secondary consumers and up towards the apex predator. So on land, you're talking about like plants and plant-like organisms, which include these guys. In the marine system, again, things like your diatoms would be functioning there as well. Anything that can photosynthesize is really quite important there. And there's some symbiont issues that can be found here as well. So here's one that was not noted to you until now. There's a dinoflagellate that's actually found in corals and some jellies. I don't even think it mentions jellies there, but that are called zooxanthellae. And the zooxanthellae actually photosynthesize material for the jellies, like maybe the Cassiopeia zamanchana, that's an upside-down filter-feeding jelly. So they photosynthesize for them, and of course for some of the coral polyps. And in return, the jelly and the coral, of course, is getting the dinoflagellate, the zooxanthellae, this lovely little home and of course, free food. It works out pretty well. It's a unique mutualism that happens there. Oddly enough, though, that when the jelly, for example, gets stressed out and something's not right in the environment or something's going wrong, one of the first things they're known to do is dump the zooxanthellae. It's like a bleaching mechanism that happens to them. So we keep an eye on that as scientists, and we see that kind of stress that's occurring. We start wondering what's happening in the environment. Is it human-induced? Is there something else going on? Which is too bad for the jelly or the corals, because of course they need these zooxanthellae for their survival. So it's a bit of an interesting issue there. And then, of course, bear in mind, no animal on the planet has the ability to break down cellulose. We've talked about that before. So we rely on other organisms as well, like the mastigote, that's helping, in this case, termites, to break down wood, this kind of issue. Of course, parasitic, we've already mentioned most of these. There's Plasmodium again. Here's the dinoflagellate and fish kills that we showed earlier. Here's oaks. Plants, again, getting hammered. Poor plants, they get hammered by a lot of things. So again, a lot of parasites are out there as well. But as I mentioned, one of the most important things is the producing system, that they can work as a primary producer. And from that primary producer, structure out a lot of the web system. And therefore, you're going from the primary producer again to the primary consumer, secondary consumer, and all the way up to the apex predator. We need them. And you even get some large organisms that are reliant on some of the lower, if you call it that, lower-based organisms. So like the zooplankton, which of course the zooplankton might be reliant on the phytoplankton. So again, it comes back a lot to organisms out there, and some of these guys might be in here, some of those phytoplankton might be in here. So that's important. Now, for the limiting factors, I noted that before, if you're talking about some of these blooms that happens, you add in one of the limiting reagents. So nitrogen, phosphorus, and iron are all limiting reagents that can be added into the system. And if that happens, you get this eutrophic condition that creates, and you get this massive bloom that can occur. And of course, it can be directly toxic, as well as create the hypoxic environments. So it's very dangerous when that happens. And although it might be natural, we got to look at human issues as well and see if we're accidentally artificially creating that. So there you go. Here's some of that issue of food chains. You can see down here some of the producers we're talking about, and then they get fed upon by other organisms that work their way all the way up to the apex predator. Here's the Ursina orca up there at the top. That, of course, not feeding directly on the zooplankton and the phytoplankton, of course, but they feed on the things that fed on them. So it's very important, again, to make sure that something as little as a protistan is out there doing its job. Without it, we can be in big, big trouble. So we see a lot of that there. And we obviously then have to worry about environmental conditions. What's allowing for these blooms to occur? What's causing crashes? What's causing populations to have problems? Of course, global warming comes up. That's always quite an issue. You know, sea surface temperature is kind of an intriguing one. We look for indicator species like corals might be having an issue. We wonder why. And things like temperature and chemistry might be affecting there. So then we trace back and say, well, has this happened before on the planet? If it has happened, what was the overall effects? Do we unfortunately get like a mass extinction that can occur? Is it something a little less serious? And of course, could that happen today? Global warming, of course, is being argued all the time. And I would postulate to you, of course, that you need to go and test this yourself. You don't need to believe what we say as scientists or what the general media says. You're educated as well, and go and read. See what's in published research. Did they do the research properly? Do you see any flaws in the research? Or was the research done well? And if it was done well, what were the conclusions? And draw your own ideas as to whether or not you think global warming is happening. Of course, we think it is, and of course, problems that we see coming out of that. But go and test it yourself and see what's out there. Okay, oops, sorry. As I noted to you, this is sort of just a nice introduction to the protistans. I know it takes a while to talk about this, and this is just the small view of it. So I still very much recommend then that you read the full notes I have for you online. Of course, that's what I'll be using for your exam as always. And then, of course, to supplement that with a textbook. You should always be reading your textbook and asking any questions you might have with the book itself or the reviews that are from the author, whatever it may be, of course, my notes themselves, and let me know what you might need for your exam. Beyond that, though, thank you very much. I appreciate it. I know it takes a while to get through these, but come on by if you have any questions, and we'll get yourself ready not just for the exam, but for our flipped classroom session. Thank you very much.